Welcome to this overview of Biobasics for Bioinformatics, where we link core biological concepts to computational analysis. In this presentation, we'll cover molecular foundations, genomics, protein structure, and cellular systems, then show how data-driven methods like sequence analysis, databases, and computational models turn biology into actionable insights for research and application. This slide shows how genomes are organized and why structure matters for analysis, with a diagram of DNA wrapped around histones forming nucleosomes, loops, and topological domains. Knowing base pair scale features, centromeres and telomeres, exons versus introns, and repetitive elements helps guide alignment, copy number assessment, and accurate gene annotation in downstream pipelines. Here we decode gene regulation from promoters and enhancers to transcription factors and epigenetic marks, illustrating the flow from molecular mechanisms to computational interpretation. The diagram highlights methylation and histone modifications affecting accessibility and shows how ChIP-seq, ATAC-seq, and motif enrichment combine to map protein-DNA interactions and quantify expression changes for integrative regulatory analysis. This slide explains protein domains as conserved functional units, using examples like kinase, SH3, and zinc finger domains, and shows how databases such as PFAM and Interpro classify them. Recognizing domain architecture improves homology inference, function prediction, multiple alignment, quality, and choice of scoring matrices, which together strengthen sequence analysis and functional interpretation. We introduce enzymes and metabolic networks by connecting enzyme kinetics, active sites, and catalytic constants to larger pathways like glycolysis and the TCA cycle. The visuals show energy barriers and metabolite cycles, while the bioinformatics applications include reconstructing genome-scale metabolic networks, performing flux balance analysis, and interpreting metabolic data to understand system-level behavior. This page covers post-translational modifications and how they change protein, state, and function, with examples like phosphorylation, glycosylation, ubiquitination, and acetylation. The mass spectrometry diagram shows how modifications appear as mass shifts, and the narrative links detection and PTM databases to site prediction tools and signaling network interpretation that drive research advancement. We review mutation types from single-base substitutions and indels to structural variants, and highlight factors that affect reliable variant calling, such as transition-transversion ratios, indel length distributions, and regional mutation biases. The slide also touches on molecular mechanisms and analytical strategies, like filtering and evolutionary modeling, to reduce false positives and estimate sequence divergence. This slide explains how selection shapes allele frequencies, contrasting purifying, positive and balancing selection, and shows common detection metrics like DN, DS, Tagemas D, FIST, and haplotype structure. Understanding these signals helps prioritize functional variants, identify potential drug targets, and interpret population genomic patterns in evolutionary and clinical contexts. Phylogenetic analysis is framed as modeling evolutionary relationships using trees, branch lengths, and substitution models that correct for multiple hits. The slide highlights methods like maximum likelihood and Bayesian MCMC for estimating topology and timing, and emphasizes how understanding model assumptions and bootstrapping produces robust trees for orthology, dating, and comparative analyses. This page compares sequencing technologies and analytical strategies, contrasting short-read platforms like Illumina with long-read PacBio or ONT, and chromatin confirmation assays like Hi-C. We cover strengths, characteristic error profiles, and file formats, and stress that matching technology to the biological question guides, experimental design, pre-processing choices, and quality control thresholds in the pipeline. We discuss multi-omics layers and library strategy, describing how RNA-seq quantifies expression and splicing, while bisulfite and ChIP-seq map methylation and histone marks, and how integration reveals regulatory influence on mRNA output. Library type choices, poly-A versus total, single-end versus paired affect alignment, normalization, and downstream statistical testing and interpretation. This slide walks through bottom-up proteomics and metabolomics workflows, from protein digestion to MS spectra and peptide identification, and from GCMS, LCMS, small molecule profiling, to retention time calibration. It shows how ion counts and database search scoring feed into pathway mapping, and how combining modes yields systems-level insights through multiomics integration. Here we outline how to integrate biological data for network modeling, listing input types like genomic variants, expression levels, protein interactions, and metabolic flux, and showing how unified network models feed tools like string, reactome, and genome-scale metabolic models. Guiding principles include identifier mapping, mitigating batch effects, and applying network inference algorithms for context-specific prediction. The final slide connects bioinformatics applications to clinical and regulatory frameworks, showing how integrated molecular profiles enable patient stratification, drug response prediction, and adverse event anticipation. It also describes pipelines that translate genomic data into actionable reports and emphasizes regulatory standards, data privacy, and evidence levels needed for responsible deployment of computational diagnostics and therapies.